So hello and welcome to the CSF Rheumatology Author Interview Podcast. I'm Professor Peter Nash from the Griffith University in beautiful downtown Brisbane. And today we're very lucky to be joined on his first day of his vacation, Professor Axel Finckh from the University of Geneva. Welcome, Axel, and thank you so much for giving up your time. Today we'll be discussing a real-world observational study which evaluates drug maintenance in patients with rheumatoid arthritis in Switzerland. So Axel, first, would you mind just telling us a little bit about yourself, your career, and how you wound up where you are and what you're doing, so the audience can get a bit of a background for you as an author?
Yes, hello, Peter. Well, my name is Finckh. I'm a rheumatologist, a clinician mainly. I did an education in epidemiology, and so in my research I work a lot with observational data and do pharmacoepidemiology as part of my research.
So that's great. So we're talking about a paper that you published in RMDO, which is a comparative effectiveness of anti-tumor necrosis factor agents, biologics with an alternative mode of action, and tofacitinib in an observational cohort of patients with RA in Switzerland. So let's just start. Could you tell us a little bit about the Swiss registry so we can get a feel for, do they capture all the RA patients in Switzerland? How does it work?
Good question. So yeah, it's a population-based registry, so meaning it is trying to cover the whole Swiss RA population, which obviously is impossible. What it probably catches very well is patients with severe RA, meaning those patients that require targeted therapies. In order to get reimbursed, you in theory at least, the insurances want you to be in the registry in order to verify that the therapies are used correctly. So we capture a good part of patients on targeted therapies, probably only part of the patients that are on conventional DMARDs.
And just roughly, how many patients in the registry and how many years have you been going?
Oh, this is an old thing now. It started in 2017 in Zurich. There are about 15 to 20,000 patients. Not all of them are active by far. So it's a fairly large registry. And the interesting part is that the biggest proportion of patients does not come from universities or teaching hospitals, but it comes first from private practices and from regional hospitals, non-academic hospitals.
And is there a control group of any kind in the registry, or is it all patients on biologics and TSDMARDs?
No, still about 50% of patients are on CSDMARDs, conventional synthetic DMARDs. And so the other half is on targeted therapies. This changes obviously over time, but roughly this is the numbers.
And one of the things that we've had issues with the Corona registry and others is how kind of trustworthy do you take the assessments of disease activity, and how complete is the data set? Do you have lots and lots of missing data?
Right. That's a good question. You know, even if things are reliable, measuring a disease activity is actually quite variable between doctors, right? If you ask the same patient to be evaluated by 10 doctors, you won't get exactly the same results, far from it actually. The reason we measured as our primary outcome in this study drug retention is that this is the outcome that is most reliable. Doctors at least input the treatment start and treatment stop, that will do. And they will report disease activity, but I think it's less reliable than treatment maintenance.
And just give us a rough idea of the Swiss market. what percentage might be on a TNF, roughly, what percentage is on a JAK,
Right.
just so we get a feel for the distribution.
It is lower than, for example, the US or Norway, but there would be about 20% of RA patients are on targeted therapies, just in a rough estimate.
And of
Large
those
proportion,
20,
but not
just
huge.
to feel for what percentage on a TNF, what percentage on tocilizumab, what percentage on JAKs.
Okay, oof, that's a difficult one. It's changing over time, right? My feeling is that currently still about 2/3 are on a TNF historically and have been for a long time, and roughly 1/3 will be on other modes of action, biologics, and now perhaps 10% on JAKs, JAK inhibitors. The JAK inhibitors are increasing a lot and fairly fast, but I can't give you the exact numbers, sorry.
Well,
Well,
no problem.
I do actually, because in the study, we took patients who initiated these therapies between 2013 and 2018, I think. So there were a total of 4,000 treatment courses. And on these 1,860 were on TNF, 1,400 roughly on other modes of action biologics. 1,000, excuse me, I need to start again. 800 initiated tofacitinib, 1,860 initiated TNF inhibitors, and 1,350 other modes of action via biologics. So it's actually a bit more. So it looks like it's about 30%. The JAK inhibitor we had at that time was only tofacitinib, and close to 50% TNF, and another 20% on biologics of other modes of action.
Thanks very much. Thank you. Did any biosimilars in the Swiss market yet? Has it made any impact, or did you just combine them under the TNF umbrella? Or the other umbrella?
Yeah, we just combined them on TNF inhibitors. But there are biologics, yeah, biosimilars. I can't tell you exactly the proportion.
Okay. So can you tell us a little bit about the study, what you tried to do with this study and what your primary outcome was?
So we basically took this period when tofacitinib was also available, so starting 2013. And we looked at how long people were keeping their drug maintenance, drug survival, or however you want to call it, as a measure of overall satisfaction. It's a mix, drug retention, a mix between effectiveness and patient and doctor satisfaction and tolerance, safety. The idea being that somebody who's happy with his treatment and tolerates it well, he's going to keep it. If either it doesn't work out well or you have an adverse event, well, you're going to stop it. We compared the drug retention between these three large groups. Tofacitinib on one side. We did not include baricitinib yet because it just came out. We didn't have enough follow-up. TNFs with all the 10 different brands and other modes of action biologics. We excluded rituximab here. Rituximab is so difficult to measure when people stop it. Some people keep it every 2 years and you think they've stopped. No, they will say, I'm still on that medication. So we thought we only look at abatacept and tocilizumab and other anti-IL-6.
Okay, and tell us a bit about what the drug survival looks like on the different treatments.
Yes, so this is actually the main results, right? So with just one word of introduction, drug retention is very country specific, strangely enough. But if you compare drug retention, I don't know, in Eastern Europe and in Switzerland or Norway or the US, you will find huge differences. It has to do with availability of the drugs and to do with the easiness. I don't know if that word exists. People can switch drugs. If it's very easy to access them, well, doctors and patients will just try out different things quite frequently. So in Switzerland, the fairly short drug retention rates, but that's just reflecting the facility of access.
Okay, so so can you just tell us what the main findings of the study were then?
Yeah, the main finding was that the drug retention or drug maintenance or drug survival was actually very good with this new drug, tofacitinib, compared to the established drugs. So on average, tofacitinib was maintained 1.9 years compared to 1.7 years for other biologics and compared to 1.3 years on anti-TNF. If you do the statistics on it, it was significantly superior for tofacitinib compared to TNFs. And there was a trend, but not significant compared to other modes of actions.
Can you tell us a little bit about the combination with conventional synthetic DMARDs and the effect that had on the results?
Yes. So as you would expect, together with TNF agents, anti-TNF agents, you get most combination with methotrexate, 64%. With other modes of action biologics, it was 53%, and with tofacitinib, it was 48%. Interestingly, the impact on drug retention was neutral for tofacitinib and for the other modes of action biologics, meaning it didn't change anything. But as you would expect with TNF agents given in monotherapy, it decreased significantly the drug retention when you give it monotherapy.
You may not have the data, but when you looked at the TNF group, was there a difference between the monoclonals and the fusion protein, etanercept versus infliximab? Do you have that data? Because we don't use a lot of inflix in RA anymore.
Right. You're right there. I think it was only about 10% of patients were infliximab. We did not look at this here. We did look at this previously, and indeed the combination is most useful for infliximab or monoclonal antibodies, a little bit less for etanercept.
Okay. And you wouldn't have had enough numbers to tell us if, say, leflunomide and the TNFs improved survival. It's often mentioned that immunogenicity reduces, and if you can't take methotrexate, at least take leflunomide.
Right. No, we didn't look at that specifically. We had a group called combo with CSD-MARS, which obviously included a big chunk of leflunomide, and that was 6% on TNF, 6% on other modes of action. Interestingly, more with tofacitinib, where this was up to 20% that had some sort of other mode of action.
Okay, and we're seeing about 30% of our TNF patients on monotherapy, whether we suggest it or the patient just does it. What's the percentage in your study?
30% of patients who take monotherapy?
TNF monotherapy.
Yeah. Well, officially here, declared by the doctor, so it's probably much higher than what they actually tell us. The percent of people taking monotherapy was about a third on anti-TNF, same thing, 40% on other modes of action, and about 50% with tofacitinib.
So it's quite consistent around
that by 10 to 20%.
But still consistent across the planet. I also noticed you've got a lot of seronegative patients, like 30%.
Yeah.
In our country, a lot of that is sort of viral arthritis, but I'm not sure you get a lot of that in Switzerland. So is that standard to have 20-30% seroneg?
Yeah, that would be very standard here. In most publications, you find about 1/3. These are very established RA patients. You know, they had 10 years of disease duration, erosions. So I think it is true RA.
Okay, that's nice. So we're getting TOFA lasting about 58% drug retention at 12 months in our registry. What was the sort of yearly dropout rate, if you like, for the TNFs, the other BDMARDs, and maybe TOFA over 12 months, if you happen to have those numbers?
I need to look at the graphs to give you a rough number, because we made hazard ratios and median drug retentions. If I look at overall drug discontinuation in 1 year, it's about the same numbers, right? More than 50% have stopped. therapy
50,
within the first 12
60%.
months. Yeah.
And the other thing I noticed from the British registry is that if you switched because of adverse events, you were much more likely to switch because of adverse events to the second and third drug.
Yeah.
If you switch because of lack of efficacy, you're also much more likely to lack of efficacy for the rest of the treatments. You don't have a feel for how that came out in your study?
No, we did not analyze specifically the reason for stopping the previous drug, and why they went on the new compound. No, we didn't look at this here.
Okay. And you did some regressions and analyses. Any patient characteristics that are associated with drug survival?
Yeah, many actually. Well, line of therapy is an important one. Let me give you the complete list. Higher number of previous BDMARD, so that's line of therapy, is associated with shorter drug retention. Shorter disease duration was associated with discontinuation, strangely enough. Higher BMI values, and more recent treatment initiation. So meaning that there's a sort of a calendar year effect. In more recent years, people start to switch more than they did 10 years ago.
Right.
So that's what about it.
It'll be interesting when the other JAKs get into your registry, because we are seeing a bit of switching between JAKs, even though there's very little evidence.
Right. We looked at that, and after baricitinib became available, the hazard ratio for stopping tofacitinib was increased by 70%. Yeah, there is a strong effect of.
That's very interesting, isn't it? I wonder if that's RA beam driven. the first ever superiority, yeah.
Yeah, or was it just the the convenience of having a once daily drug that people like? I don't know.
So, what do we think about the take-home messages from your study?
I think it would be very conservative, given the observational nature. I think what we can say is that overall these drugs are maintained fairly well, suggesting that they have a good balance between safety and efficacy, and that they are at least as maintained as long as other drugs that we're used to. Probably even slightly better in this setting, meaning patients who already have experienced one previous, mainly anti-TNF drug. The other interesting thing, I think, is that methotrexate doesn't seem to be associated with longer drug retention, suggesting that perhaps the combination of methotrexate is a little bit less needed with these newer drugs than it was with anti-TNF.
Yep. And disease activity, the CDI data?
Yeah, we did measure disease activity at 12 months. As mentioned, that is a bit more difficult because not everybody measures disease activity at 12 months. But when we do that, it was very comparable between these three drugs. We did not see any significant difference between any of these groups.
Okay. So thank you very much. We thank you again for your time. Any take-home message, any last comments you want to make about this study and where you're going to next and what your next plan will be?
Well, I think safety is what people are interested in, what we are interested in. As you know, safety requires big numbers if you want to do something meaningful. So at the moment, we're working on the combination of registries, 18 European and extra-European actually registries, where we combine these data and try to do this kind of analysis, but on safety.
Well, I think it's critical because only well people get into clinical trials, and the patients in your registry are full of every comorbidity that never gets into a clinical trial.
True.
So safety is critical, but drug retention is a nice surrogate for doing well and not getting adverse events. So congratulations on your study. We thank you again for your time. We appreciate you giving up your first morning on break for us. This has been the CSF Author Interview Podcast. If you'd like to know more about this paper and others uploaded to the CSF website this month, You can get detailed slide sets available in the publication section. Just go to cytokinesignaling.com. And please subscribe to this podcast on iTunes or wherever you get your podcast from, and let us know what you think. We'd love some feedback. So thank you very much, and all the very best.